Okay, I'm going to start talking about water regulation and how animals take care of, basically how the kidneys work. But we're going to kind of use some examples of animals that don't have kidneys, and how water regulation happens in those animals in a general manner, and then ultimately get to kidneys. Now, remember, when I say water regulation, I'm not just talking about eliminating water from the body or taking in water, but the fact that we're getting rid of nitrogenous waste. Now, when you lose weight through, not eating enough calories and exercising and all sorts of things, the way that you lose most of that weight is actually breathing it out. So when you break down sugars or fats, you're breaking that down to carbon dioxide and breathing that out ultimately as waste in that way, carbon dioxide waste. However, when you break down proteins or other types of nitrogenous waste, it is done through the breakdown of those proteins into urea. And then that urea is eliminated through urine. So that's the big thing about getting rid of urea or uric acid or whatnot, depending on the type of animal, is through water regulation and so forth. And that again involves the kidneys. Obviously, the kidneys are going to be important for getting rid of lots of poisons and toxins too that you might have taken in after being metabolized by the liver. The urine is essentially blood plasma that has been concentrated in the kidneys and eliminated as nitrogenous waste, extra salts, whatever the case may be. The reason why your urine is yellow actually is due to the fact that your kidneys have broken down hemoglobin into bilirubin. And some of that bilirubin, while it might be used for bile, some of it is actually eliminated in your urine and gives it kind of that yellow appearance. Now, not all animals, as you know, would have kidneys, as you can imagine. So the simplest animals like our sponges, again, is kind of our classical example of the simplest animal, won't have any kind of special excretory system or organ system. And all the cells themselves are going to excrete directly with the environment. And that is true for a lot of animals. They may get rid of a lot of different types of waste through their skin directly into the environment. Heck, even our own skin has a little bit of urea on its surface as well. Again, remember, this is going to be floating around in your blood as your blood plasma and then get concentrated in the kidneys to eliminate that waste product. And again, it's also about regulating salts and water and osmosis and osmoregulation. The kidneys are critical for that, as well as other tissues, depending on whether you're a very simple animal or not. As you know, life has evolved in the ocean initially. And so a lot of it was about regulating salt concentrations and so forth. And water obviously moves in the direction where the solutes are, which would be your salts, across a semi-permeable membrane. So a big part of that movement of water is based on its osmolarity. And that again is the number of moles of active particles per liter of solvent. And you can watch, I recommend watching a video on that if it's a little bit confusing to you, but basically the solutes again are going to be the particles that are floating in the water, or the solvent, which is water, and that makes up a solution. So a 1 molar solution of glucose equals 1 osmolarity, and 1 molar solution of sodium chloride is 2 osmolarity. So because there's those 2 different atoms involved. Or 2 different, because they break apart, as you know, they're held together by ionic bonds, while glucose is a single molecule. So high extracellular osmolarity dehydrates a cell. So that, in other words, if you have lots of salts on the outside of the cell, water is going to be pulled away from the cell towards the solutes. So if you have a nice, healthy, round cell with plenty of water and you put a lot of salts around it, the water is going to leave the cell, making the cell shrink up. It could make it very bad for the cell because it can result in denaturing of proteins and the cell not having proper metabolism and ultimately dying in that kind of extreme environment. You can see putting an egg in salt denatures and pulls the water out of it. So water, again, going across a membrane would leave if it's more water on the inside, it's going to leave the inside of the cell and go to the more concentrated area where the osmolarity is greatest. Likewise, if the water on the outside of the cell doesn't have salts in comparison to the inside of the cell, water is going to go into the cell, causing a hypotonic solution, and the cell could burst. And so sometimes people have done studies where they look at eggs and they put them in these different concentrations, and you can see that happening. And so that might be a good thing to do. Take an egg, remove the calcium, put it into a solution of salt or a solution of basically water, and watch the egg get bigger or smaller as a result of it. The fish have to deal with this. If they're fish living in a very salty environment like marine fish, they have to make sure that they're eliminating lots of salt and maintaining as much water as possible. While if it's a freshwater fish, the water's going into the fish, and so they got to control holding the salts in relationship to the environment. So a freshwater fish is going to actually urinate a very watery pee because there's so much water coming into the fish. While in a salty environment, they're losing water to the environment, so they got to figure out ways to keep that water while getting rid of salt. So you'll have marine fish will usually have very concentrated urine, getting rid of lots of salt while maintaining as much water as possible. That's why you can't just dump a freshwater fish into a salty environment and vice versa. Most of the time they will die because they have not evolved the ability to deal with those differences in salt concentrations and so forth. There are some fish like estuarine fish so that can live in those environments where it gets salty and less salty based on the amount of fresh water that comes into kind of a backwatered marsh near the coast of Florida or something like that. But there's different ways that animals have evolved to try to control the amount of water regulation, and of course, get rid of this nitrogenous waste. And it's done so through excretory organs, like the kidneys, but there's other types. Excretory organs control the osmolarity and the volume of tissue fluid by excreting excess solutes and retaining others that are valuable or in short supply. Terrestrial animals also use excretory organs to eliminate waste products such as nitrogenous waste through nitrogen metabolism. Breakdown of proteins is a good example of that in their urine. Organisms have a diverse excretory systems depending on the environments in which they live. In all cases, water moves by simple diffusion. So again, it's going to move from an area of higher concentration to an area of lower concentration. The basics of excretory system functions are the same for many animals. So the way it works is basically the same, irrespective of whether they have kidneys or proteinuria, kind of flame cells and things like that that I'll show you. Or sometimes they use gills to do it in case of fish. But these excretory organs filter tissue fluid to produce a filtrate that does not contain cells or large molecules such as proteins. The kidneys, for instance, filter your blood, but you maintain the blood cells, and the blood plasma gets filtered and then, of course, concentrated. So that's the filtrate. This often is done on a blood plasma driven across the capillary walls by blood pressure. So that again is animals that have, of course, a closed circulatory system. Open circulatory system, it's a little bit different. Closed circulatory system, of course, are blood vessels and so forth. And be sure to check my blood discussion if you might have forgotten that. The filtrate is then processed usually by a series of tubules that go up and down, which happens, which you'll see in the kidneys as we get to that part, where active secretion and absorption takes place that ultimately changes the composition of the filtrate. In some cases, it makes it more concentrated, and in some cases, it makes it less concentrated. Even you've seen this in your own self. If you drink a lot of water, your urine's going to be less concentrated than if you haven't been drinking water. Some animals can kind of like change their salt concentration to what it's like in the environment. We call those osmoconformers, where the osmolarity in their tissue fluids equilibrates with the external environment. It's kind of like when you think about ectothermic animals or cold-blooded animals, their body temperature kind of is relative to what you see in the environment. Well, osmoconformers, their salt concentrations are relatively equal to what's happening in the environment. So unlike us, we're not osmoconformers. So you put us in the ocean, we're not going to have a concentration of salt that's similar to the environment. We're going to lose salt um, to the environment. Now, obviously, our skin's going to provide a lot of protection, but a little bit of salt water starts getting into our mouths and so forth, and we begin to urinate more and more in those salty environments. So so again, animals, we'll get to that in a moment, but some animals are able to maintain their salts relative to the water outside. We call those osmoconformers, and we'll see example in a moment. Osmoregulators, though, however, have to maintain their osmolarity in their tissue fluid. So we're regulatory animals as well. Fish are a good example of this. A freshwater fish would be an osmoregularity where they're going to retain as much salt as possible, and they're going to have very dilute urine. If you're a saltwater fish, you're going to maintain as much water as possible while getting rid of that excess salt, as I alluded to earlier. So here's an osmoconformer. This is a brine shrimp. And you can see that its body will change the amount of salt concentration relative to the water that happens to be in. So if it's in a dilute environment, the body will actually become more hyperosmotic. It'll become, relatively less salty. Well, let me make sure I back up a little bit. In a salty environment, you can see the seawater, and it shows that it'll osmoconform in seawater to a certain degree based on the amount of salt in it. When it gets to extreme hyperosmotic situation where the seawater is very dilute, maybe in an estuarine environment, it's rained a lot, the fish then has to start urinating the brine shrimp will begin having to urinate very watery pee. But if it goes into a hypoosmotic environment where the pond becomes more and more salty, for instance, it's going to have to hold on to more of that water and get rid of extra salt. So in the middle, where it's not too salty or too dilute, the body will actually change its salt concentration based on amount of salt that's in the environment or in the water. But again, once it gets into that really salty pond, it's going to get rid of more salt and hold on to water. And vice versa, the pond becomes very dilute, it's going to pee more watery. So there's a point where it becomes an osmoregulator is what I'm trying to get at. It's osmoconforming, and then it gets to these extremes, it becomes osmoregulating. And so that's what's mentioned in this. And most of this is done through the gills. So the the shrimp will, again, can secrete out chloride and sodium and take care of all that kind of regulation through their gills. In some cases requiring active transport. You can see this also in marine fish, like our seagulls. They can get rid of excess salt through nasal cavities or through their eye. So here we have terrestrial animals will get rid of excess salt, but that's not usually a problem because most plants are low in sodium. So herbivores must actually conserve their salts. That's why you see deer at salt licks, because there's so little salt in the natural environment. So if you sprinkle salt on the road through de-icing, a lot of times you'll see white-tailed deer just eating salt right off the side of the road. So terrestrial animals are trying to maintain that extra salt usually. While birds that are marine birds get rid of that excess salt because they live in a marine environment. They're eating fish with salt in it, they're drinking bits of salt water. They got to get rid of that excess salt. And they do so through nasal salt glands. So they'll get rid of the concentrated salt through their nasal salt glands. And so here you can see um, a central canal that pulls in that excess salt right from the capillaries and into the gland, and then ultimately it'll come out as a droplet of water that's extra concentrated in salt. And so here you can see one of the examples of a seabird that can do that. So the big thing about terrestrial animals is we need to use our kidneys, and this might be true for marine life as well, but we need to use our um, ability to excrete watery waste to get rid of nitrogenous waste. Now remember, carbohydrates and fats are eliminated through breathing out carbon dioxide. So we'll get rid of carbohydrates and fats by breathing out carbon dioxide through, again, cellular metabolism, cellular respiration, and making ATP. Now, again, you'll take in protein that you'll eat. You'll be digested, made into human protein, but often a lot of that will be secreted as well. Or if you lose proteins in your arm, maybe from wasting away, you're going to get rid of protein that way as well. But your urine is important for getting rid of proteinous waste. It'll be through the breakdown of proteins into amino acids, because that's the building block. Proteins is amino acids. The amine group is NH2. Nucleic acids also have nitrogenous bases, and that leads to these amine groups. Now, some animals will get rid of this nitrogenous waste through ammonia by adding an extra hydrogen to it, and we call that ammoniotelic animals. We animals, for the most part, will get rid of nitrogenous waste by urea, so that we are urotelic animals. That doesn't mean there won't be a little bit of ammonia in our urine, though, too. But this is an efficient way to get rid of nitrogenous waste without losing as much water. Fish are an example of animals that would more likely follow the ammonia or aquatic freshwater animals, because there's so much water around. They don't have to do this more efficient way of getting rid of nitrogenous waste that conserves water, like urea. Now, some animals, like birds, will mostly get rid of their nitrogenous waste through uric acid. So it'll be a very hydrophobic water or waste product. This is the way to conserve the water the most. And this also leads to bird droppings and why they're so hard to wash off of your car, because uric acid in their waste, which of course includes feces as well, will just stick to your glass, and it's hard to wash off because it's a hydrophobic substance. So this is the way that these animals conserve the most water. Now that doesn't mean, again, that you don't have uric acid and have the ability to make uric acid, which isn't always a good thing for us. So urea is the ideal way to get rid of nitrogenous waste. Again, ammonia is very toxic. It works well more in a freshwater environment. Urea and uric is less toxic and less hydrophobic. It requires more metabolism. So aquatic animals continuously excrete ammonia. That's one of the reasons why you have to change their fish tank, because they've been peeing in their fish tank. That ammonia is toxic to the fish over time. It's very soluble and diffuses rapidly across the gills. These animals are, again, ammoniotelic. You may want to double check my pronunciation on that. In mammals, though, we are ureatelic animals. And so we're able to excrete most of our waste as urea, or in some cases, particularly if you're a bird, uric acid. That doesn't mean there isn't a little bit of ammonia also in our pee as well, but it's around 5 milligrams per 100 mils. Remember, that's enough. Well, excuse me. That little bit of ammonia is enough to be lethal in our body. So that's one of the reasons why urea is running around in our blood versus ammonia. Urea is water soluble, and so we've evolved to get rid of it by concentrating it in our kidneys. Some fish actually, though, like our cartilaginous fish, cartilaginous fish like sharks and skates and rays, will actually hold on to that urea in their bodies that helps them to maintain um, their osmotic regulation. And I understand that you need to wash sharks and fish to get rid of urea from their muscle tissues before you eat those type of fish. Again, insects, reptiles, and birds are more likely to be urotelic as well. So that means they're going to be making uric acid. Uric acid is water insoluble, is excreted as a semi-solid, whitish. That's the whitish material you see in bird droppings or insects. Uric acid is the best way to prevent water loss during the removal of nitrogenous waste. That makes a lot of sense why you'd want to be an insect, particularly a terrestrial insect, and use uric acid because you would run out of water and become dehydrated rapidly, or birds. Some amphibians do it too, but most amphibians that live in the water will also do ammonia. Now, most species produce more than one nitrogenous waste, and that includes us. Humans are ureatelic. That means we're making more urea, but also excretes a little bit of ammonia and uric acid in their urine. Different developmental stages may also form different types of nitrogen excretion. For example, tadpoles will excrete a lot more ammonia when they're living purely in that aquatic environment than adult frogs who come out on land and need to conserve that water, and they'll excrete more urea. Some frogs and toads in arid regions also excrete uric acid because they got to maintain as much of that water as possible. So there is some flexibility in the type of nitrogenous waste as terrestrial animals will excrete. And that probably is true for a lot of aquatic animals as well. Now you can see an early kidney in flatworms, Planera. They excrete water by means of a network of tubules that run through their body. Each tubule ends in a flame cell. So you can see a flame cell at the end of it right over here in this picture. And so it's constantly spinning and pumping water through their system. And then the nitrogenous waste will go, and whatever waste that needs to be gotten rid of, will go through these tubules, because remember, they don't really have capillaries or anything like that, into tubule cells, and then excreted out the excretory pore. So this is kind of what a primitive kidney would look like. Again, we call them flame cells. They have cilia that are constantly beating. The flame cells in the tubule together form a protonephridium. Now remember, proto means early, so nephridium is kidney, so it's kind of like an early primitive kidney. That's what it's basically saying. These tissue fluids enter the tubules, and beating cilia cause this fluid to flow toward the excretory pore of the flatworm. And then the fluids can be modified, concentrated or less concentrated, based on what needs to be gotten rid of and what kind of environment there are. If they're in a very watery environment, they're going to try to conserve ions and excrete more water. If they're becoming in a more dehydrated environment, likely they will try to conserve that water. We begin to see a more complicated kidney in earthworms. That's also where we start to see blood capillaries, if you remember that from some previous talks. These are found in worms, earthworms specifically, not nematodes, earthworms, known as annelids. They have a closed circulatory system, so that means they have arteries and capillaries and veins and so forth. 1 second. Okay, so annelids have a closed circulatory system. And so the blood is filtered through the capillaries and so forth. The fluids are running around in the coelom also. And so what happens is the capillaries are filtered through a metanephridia. Again, it's going to be a filtrate. So the blood is going to be filtered into this capsule called the nephrostome. So you can see in this picture here, and I'm pointing to it. Let me get back into presentation mode. So you can see the nephrostome. The kidneys are essentially, that's like a kidney, and there's little guard cells and things like that. And so as the capillaries are going through it, the blood is being diffused from the capillaries into the nephrostome. So it's actually concentrating the urine. And then the urine will enter into the nephrostome. And then depending on the coelom, it'll either become more diffused or less diffused, depending on whether they're in a hypotonic or hyper, depending on the environment they're in. And then the water will be filtered out of their blood. Obviously, I imagine on a very rainy day, they have to come up to breathe and, that have muck or they'll die. I imagine at that point, their urine is very dilute. And then, of course, when the soils become dry, their urine must become more concentrated. And so the kidney system, this metanephridia, is controlling that. Again, their blood's going to be filtered into the nephrostome, the watery part of it. The blood cells or whatever will remain in the capillaries. And then the tubules itself will help to concentrate or not concentrate it based on the coelom, the watery stuff that's running around in the earthworm, the empty space around it. Now, the kidneys of insects are called Malpighian tubules, named after this Italian science Malpighi. And the Malpighian tubules are filtering the hemocoel of the insect. The hemocoel, they don't have capillaries and blood vessels, so they have an open circulatory system. Insects do. And so where do you find the Malpighian tubules? Well, they're between the midgut and the hindgut. You'll find these tubules coming off of it. And so you can see the Malpighian tubules. So they'll eat food. The food will travel down their gut, down the midgut, down to the hindgut where it becomes feces, kind of like the large intestines and so forth. The midgut's kind of like the stomach and small intestines. And so right between them is the Malpighian tubules, and they're just floating around in the blood, concentrating uric acid from metabolism that took place, the nitrogenous waste metabolism. And so their nitrogenous waste and fecal material will leave through the rectum, and we call that frass. So here's our Malpighian tubules filtering in sodium, which is going to draw water in, and uric acid in, and then ultimately that will go down to the hindgut, and then from the hindgut, right before it's gotten rid of, the insect will reabsorb as much water and salts as it can or as needed for the metabolism, and the uric acid and whatever they ate will be eliminated. Technically, that feces is called frass in insects, by the way, frass, F-A-R-A-S-S, and it'll look like crystals of uric acid. Sometimes we'll get uric acid from unfortunate metabolism of nitrogenous waste in our muscles, and that causes gout and the muscles hurt really bad. That's a disease where we might use uric acid, unfortunately, in the wrong way. Just remember, Malpighian tubules are used for insects. So now we're going to stop here, and then in our next lectures, we're going to start talking about kidneys and how they work in vertebrate animals, animals with backbones. So fish, cats, what I'm talking about, humans. We'll use humans as the model though. Anyway, thank you for your attention, and that's our intro to talking about how kidneys work. Have a good day.